So now the muscle has been excited from the neurotransmitter acetylcholine. The next step is excitation-contraction coupling. So before that, the end plate potential that's generated causes this local wave of depolarization. So again, it's this graded potential. So now the resting membrane is polarized, meaning that there's a voltage difference between the inside and the outside, more than normal. So the next step is because of those graded potentials, the end plate potential, there is an action potential that can occur. So that end plate potential, the local depolarization, can lead to depolarization, which is the beginning of an action potential. The second part of an action potential is the restoration of the resting conditions. So we're going to look a little more closely at these gates now. So before, when we talked about the acetylcholine gate, remember that that was a chemical or ligand gate. This one is a voltage gate, and it's open for only one ion, which is sodium. So when sodium enters the cell, that is the first phase of the action potential called depolarization. Because the inside of the cell goes from its resting membrane potential, which is negative 70 millivolts, to plus 30 millivolts. So the depolarization is the generation and the propagation of an action potential. So sodium enters. After the sodium enters, then potassium is going to exit. And when that occurs, that's called repolarization, which is what we see down here. So the potassium channel opens and only potassium leaves for repolarization. And now the inside of the cell goes from positive 30 millivolts back down to negative 90 millivolts, and then it returns to negative 70 millivolts. And that'll make more sense in a couple slides when we see the graph of the action potential. So this slide is showing excitation contraction coupling, which sounds like a lot, but remember, it's just putting two things together. It's putting the excitation of the muscle together with the contraction of the muscle. So now the action potential is propagated, meaning it travels along the sarcolemma and into the T tubules, which remember is simply an extension of the T tubules. And in the T tubules, there are voltage sensitive proteins, and these voltage sensitive proteins are going to allow calcium to be released from storage. And it's that calcium that leads to contraction. So here's what we've kind of seen. Excitation contraction coupling is the sequence of events by which transmission of an action potential along the sarcolemma leads to the sliding of myofilaments. So we've discussed the events at the neuromuscular junction. Now, after the acetylcholine binds, there's a local potential, but that leads to an action potential generated. So I want to make one thing clear here. Some students get confused because this was an action potential in the motor neuron. So I like to think of that as action potential number one. This is a different action potential that's now generated in the sarcolemma, and it's going to lead down into the T tubules, and eventually calcium is going to be released into the sarcoplasm. This is going to show an enlargement of just this area right here. So we're going to zoom in again to see that, and we see the sarcolemma here. The sarcolemma is where this second action potential is now continuing down into the T tubules. Once the action potential gets there, voltage sensitive proteins are going to cause calcium to be released into the sarcoplasm. So calcium ions are released. Once they are released, they can then bind to troponin. So you make sure you know it's troponin and not actin or tropomyosin. Troponin's connected to tropomyosin, so as a unit, both of them rotate out of the way to uncover the binding site for myosin. Because eventually myosin, which remember is the thick filament, is going to attach to the thin filament, which is actin, to form a cross bridge. So the calcium binds to the troponin, the myosin binding sites are exposed. Now myosin can grab it. So again, if we think of this like a hockey stick, this would be the part of the hockey stick that comes in contact with the hockey puck, and specifically it grabs or attaches to the actin to form a cross bridge. And that cross bridge cycle we'll be talking about coming up. But remember, it's that third video that you want to watch in the A&P Flicks section. So once the action potential ceases, calcium then is going, the voltage sensitive tubules return to their original state. But basically, calcium levels in the sarcoplasm fall because they go back into storage in the sarcoplasmic reticulum. Therefore, without calcium, tropomyosin is restored, and there's no cross bridges that are formed. So each time an action potential arrives at the neuromuscular junction, the excitation-contraction coupling process is going to be repeated. So let's take a quick look at the graph for the action potential. An action potential is what we call an all or none signal. And what that means is that all are exactly the same. So they have the same amplitude. So graded potential, those are different. That's the end plate potential. Not all of them are exactly the same. So when a cell is at rest, the inside of the cell is more negative than the outside. So when they put an electrode to discover this into the inside of the cell, and they discovered that compared to the outside of the cell, the resting membrane potential was negative 90 millivolts. Sometimes it's negative 70, but in this case it's negative 90, which is the case for a lot of times nerves and muscles. But the threshold is negative 55 millivolts, which means once enough sodium enters the cell, then an action potential occurs. So negative 55 is the threshold. And the first thing that you saw was that voltage-gated sodium channels open, and that causes depolarization due to sodium entry. So the inside always goes from negative 90 to positive 30. Once it gets to positive 30, those voltage-gated sodium channels close, and the voltage-gated potassium channels open. And this leads to repolarization because of potassium exiting the cell. So it's almost like the inside of the cell then returns to its normal polarized state, where the inside is more negative than the outside. So that leads to the inside becoming more negative all the way back down to negative 90.